Determinants of cortical size. The size of the cortex varies greatly among mammals, but it is enormously enlarged in primates. This growth has not been homogeneous and has consisted in a great increase in surface area and a little increase in thickness. The surface area of the human cortex is 1,000-fold larger than that of the mouse, but its thickness is only about 2 or 3 times that of the mouse cortex. This asymmetric growth seems to be related to the anatomical and functional structure of the neocortex that has been described as formed by iterative units named radial columns, cortical columns, or cortical modules, consisting in a columnar array of neurons that share a common basic scheme of intrinsic and extrinsic connectivity and subserve the same function. The larger the cortex in a given species, the larger the number of participating columnar units. The liberal use of the broad and undefined term cortical column has caused considerable confusion in the literature, since there is not a unique anatomical or functional columnar unit that applies to all cortical areas or species.
Columnar.
Columnar unit. In spite of this heterogeneity, there is consensus concerning the idea that the cortex is composed of an array of columnar units of operation. Under this perspective, it seems that the addition of new functions to the cortex does not require a substantial increase in the thickness of the cortex, namely, in the number of components within the cortical units of operation, in spite of a very likely increase in their complexity, but would mainly correspond with the addition of new columnar units, which ultimately would contribute to the forming of new cortical regions and functional domains. In this respect, the radial unit hypothesis provides a mechanistic explanation of the large expansion of cortical surface without a concomitant significant increase in thickness during phylogenetic and ontogenetic cortical development. It also shows how the genes controlling the number of founder cells in the mosaic of proliferative units at the ventricular surface set a limit on the size of the cortical surface during the development of the individual, as well as during the evolution of the mammalian species. For example, a relatively small change in the timing of developmental cellular events could have large functional consequences. A minor increase in the length of the cell cycle or the magnitude of cell death in the ventricular zone could result in a large increase in the number of founder cells that form proliferative units. Since proliferation in the ventricular zone initially proceeds exponentially because of the prevalence of symmetrical divisions, an additional round of mitotic cycles during this phase doubles the number of founder cells and, consequently, the number of ontogenetic radial columns. Figure 1.5. According to this model, fewer than 4 extra rounds of symmetrical cell divisions in the ventricular zone before the onset of corticogenesis can account for a tenfold difference in the cortical surface area. Since the mode of cell division changes to predominantly asymmetrical after the onset of corticogenesis, one can predict that the cell production period in humans will be about 2 weeks longer than in macaques, but should enlarge the cortical thickness by only 10% to 15%, a result which is actually observed. Rakesh, 1995. Thus, as illustrated in Figure 1.5, even a small delay in the onset of the second phase of corticogenesis results in an order of magnitude larger cortical surface because of the increasing number of founder cells at the ventricular zone. This has been observed in mice expressing higher levels of beta-catenin that enhance the number of founder cells and can lead to formation of ectopias as well as an expanded cortical plate that begins to form convolutions. In addition, the number of cortical neurons will depend on the degree of survival of the generated cells, and thus one mechanism that regulates the number of cells produced in the ventricular zone is programmed cell death, PCD, or apoptosis. Although programmed cell death has been considered a major factor contributing to the formation of the vertebrate brain, Glucksmann, 1952, contemporary research has focused mainly on the histogenetic cell death involved in the elimination of inappropriate axonal connections at the later stages of development. However, the discovery of several classes of genes involved in apoptosis, which were initially identified in invertebrates, created the opportunity to study this phenomenon in the mammalian cerebrum. For example, a family of enzymes called caspases has been shown to play an important role in apoptosis in a variety of organs and tissues. In Ellis and Horvitz, 1991. We have demonstrated that in mouse embryos deficient in caspase 9 and 3, fewer cells are eliminated than in their litter mates. Quita et al., 1996-1998. Reduction of apoptosis in the knockout mice for these caspases results in the formation of supernumerary founder cells in the cerebroventricular zone. As a consequence, these mice form ectopic cells in the intermediate zone, as well as a larger cortical plate with more radial units. Correspondingly, diminishment of the clearance rate of apoptotic cells in transgenic mice lacking a receptor crucial for the recognition of dying cells results in enlarged proliferative zones that lead to the formation of ectopias, as well as an expanded cortical plate that begins to form convolutions. In both cases, by increase of the founder population or by reduction of apoptosis, the result is a larger cortical sheet that begins to buckle and forms incipient convolutions without significant change in thickness of the developing cortical plate. This result is a good example of how the mutation of a few key genes that control the reduction of cells could result in the expansion of the cortex and the appearance of convolutions during the evolution of the cerebral cortex.